This webinar is the second of our series of webinars around the topic of biocuration, and there will be more webinars about this topic following on the next Tuesdays until the 12th of April. Next slide, please. I would like to tell you a bit more about the background and the motivation why we run this webinar series. So in 2019, Elixir launched an implementation study, and this implementation study was designed to identify communities of biocurators, to review the work that biocurators do, to identify resources and tools that are used by biocurators in their daily work life, and to identify biocuration training needs and gaps. This study comprised a survey which was sent to biocurators and to people who work in related fields, and it also comprised workshops in which we discussed these topics with biocurators. Next slide, please. The survey, the study showed that the training needs that biocurators have are most commonly programming, scripting and coding, development and use of ontologies, and database management. The survey also showed that skills like programming, scripting, and coding, and also extracting data from literature are very decisive for a successful career in biocuration. But a lot of respondents to our survey also said that they are most difficult to learn. The survey also showed that only few training offerings are available that meet this very specific need of biocurators. And if they are available, they are difficult to find. Next slide, please. With our webinar series, we hope that we can help to close this training gap a bit. And this is why we will cover interesting topics and relevant topics for biocurators within the next couple of Tuesdays. And we have already started with this last Tuesday when we talked about programming skills in biocuration. Today, we will talk about text mining for biocuration. And then next week, we will talk about ontologies for biocuration. We will also cover working with sensitive data, and we will also discuss the role of biocuration in industry. All of these talks will be recorded, and the recordings will be added to a biocuration collection of e-learning, which will allow for continuing learning on this topic. Next slide, please. As I've already mentioned, we already had a webinar last week, and the recording of this webinar is already available on our EBI training web page. Next slide, please. And now we will move on to today's webinar, and where we will discuss text mining for biocuration. And I'm very happy to introduce you now to today's speakers. Next slide, please. I'm very happy to have Shyama here. She is a machine learning and text mining scientist working at the literature services at EMBL-EBI, and she will first provide you an introduction to text mining today. And then I'm very happy also to introduce you to Michelle. Michelle is the UniProt annotation coordinator, and she will illustrate how she uses text mining tools as a biocurator. So I'm very happy that both of them are here today, and I will hand over to Shyama. Gemma, we cannot hear you. Maybe you are muted.
Yes.
Wonderful.
You can hear me now, right?
Yeah, perfect.
So welcome, everyone. Thank you for joining. The title of my talk is Target Disassociation Extraction from Literature. 1 second. Right, you can see the next slide, I think. So good afternoon, I'm Shamastri, and I'm a machine learning and text mining scientist at EBI for EWPMC team. During today's talk, I will introduce some of the text mining buzzwords, and I will use them frequently later in the presentation. Some of the ideas are probably not available to use right now, as they are active research topics, but will certainly be useful for you to know how those techniques can greatly help you in your curation projects. I'll then move on to EuroPMC and how you can use our services to mine articles and how we use text mining behind the scenes. Lastly, I will show you how we can use text mining and deep learning to extract target associations from literature. And that's the collaboration with Open Targets. Right. One of the most common terms you will hear in text mining domain is named entity recognition. According to the Wikipedia, a named entity is a real-world object, such as a person, location, organization, product, etc., that can be denoted with a proper noun, proper name. It can be abstract or have a physical existence. There are other definitions available online. You can look at them, but I think it is one of the best definitions you will find. For biomedical domain, the real life concepts are genes, proteins, disease, organisms, or relationships or pathways and so on. It can also be interaction between the entities. In this slide, the text snippet is annotated with biological named entities. The gene protein entities are shown in blue and the diseases are in green. So you can see how we use the term named entity, biological named entity, for biological existence like gene proteins. And I hope you have a little bit of idea now what it means and what I refer to when I say biological named entities in coming slides. Next challenge that natural language processing and text mining try to address is the task of summarization. So the goal is to create a summary of a given query based on available documents. So in this slide, what I'm trying to show is that if you searched EuroPMC, if you would search EuroPMC for BRCA1 gene, you'd see there are thousands of documents. So what summarization would do is create a summary with key points about BRCA1 gene. And you can use deep learning or any other machine learning techniques or any other text mining approach to get the summary. Third branch of text mining tries to go further from the summarization and directly generate an answer to a specific question. So in this slide, our question is, which repair pathway utilizes the BRCA1 protein? So if the question answering method worked nicely, what you would do is go through the articles that are available and list the pathways, repair pathways where BRCA1 protein takes part. As you now understand different text mining and natural language processing terminologies, I'll move on to the next topic. That is how EuroPMC uses text mining. So on the left-hand side, I'm giving you some statistics about the kind of information we mine. I'll focus on the text mined annotations, biological named entities, that is. So we have our text mining annotations are coming from 24 entities. For example, gene protein, disease, cell lines, mutations, drugs, chemicals, to name a few. We have our core text mining pipeline that annotates the articles on our platform. And besides our own annotations, we also host annotations done by other organizations as well. For example, Pubdata, Enact, all of their annotations are loaded on our platform. You can take advantage of text mining through two different ways on EuroPMC platform. First one is through our search engine, where you can use a number of rules to customize your query. Secondly, you can use our annotation API to programmatically access the annotations, filter them, and post-process them even further. The next couple of slides, I will dive a little bit deep into how to use individual approaches. Here in this slide, I'm showing examples of mining articles and retrieving them based on multiple criteria, such as terms, annotation types, presence of a specific type of accession numbers. Type-based filtering is only being possible because we have the biological entity recognition done on the whole of EuroPMC. In the first example, we are looking for articles where we have the term T-cell activation along with the term human, and human needs to have entity type organism. And the last filtering criteria is that the article should have a biosample accession number. So now for the second criteria, where I'm saying term human has to be mentioned as organism, probably doesn't make too much sense because human is a word that doesn't have any other meaning. But for example, if you had the word cancer, of course, it's widely used as a disease. It's a disease term, but cancer is also a species term. So you need to know the semantic of the term that you are searching, and mentioning the type along with the word could help you to reach the right point. As you can see, from this search, we only have 22 results, 22 articles that match these criteria. In the next example, I have expanded the query by adding bioproject accession type with an OR condition. And as you can see, that increases retrieved articles. What that means is how we can query EuroPMC not only by terms like T-cell activation, but also with presence of an annotation type or combination of that. So this sort of query will be very useful for you if you are trying to find articles that also has the genomic data available somewhere. On our annotation API, you can currently base the searches on 5 main criteria. Within the annotation by article IDs here, you can select which annotation you want. For example, you can only want gene proteins for a certain number of articles. You may choose annotation by dataset like KB or Campbell. So give me articles that only contains chemical entities that has KB IDs, for example, or only retrieve annotations from a particular section. So let's say if you're interested in methods, then you can say, okay, I am looking for gene protein only, and give me the result only if it appears in the method section. For open targets annotation, that is target disease association sentences, you'll need to use either annotation by provider here or annotation by relationship. You can also mix and match them in your code. So you'll have to write your code to retrieve this data and then probably filter them further by using another set of API rules. So I hope you have some idea now how you could use the EuroPMC platform to search and filter articles. Text mining and text mined information allow you to do many of this specialized search and filtering. And you can, of course, write your own code to filter them even further or customize them. I'm no way giving you a full sort of idea, or I'm not going into details how you can use each of these facilities. But if you're interested, you can always visit our website, our platform. We have very helpful help desk. You can always ask them questions as well if you're facing any problem. So this is the part where we are collaborating with Open Targets, and for them, because Open Target is interested in drug targets, we are currently providing them entity associations. So what we mean by that is we consider entity co-occurrence in a sentence as entity association currently. So association can be negative or positive or neutral. We are not currently annotating that. As part of our collaboration with Open Targets team, we use deep learning-based biological named entity recognition. So that's a method that we use to identify terms like activating transcription factor 5 or malignant glioma. We also apply deep learning approach to the evidence string. So here, the first sentence is evidence string because, as I said, co-occurrence of entities are considered as an association. So here in this sentence, a protein name and a disease name co-occur in this sentence, hence this is an evidence string. Whereas the second sentence, as you can see, only the gene name occurs, hence it's not an evidence string. So we apply deep learning approaches on these sentences to improve the accuracy of association identification. You can query these associations on our platform as well, as I mentioned before, through the relationship or the provider facility from the annotation API. Following up from the previous slide, if you are interested in, I'll tell you how to find these association sentences. So if you're interested in ATF5 protein and malignant glioma and want to find the results that we have produced from Open Targets, then you can search EMBL-EBI frontend like this. So let's say this is your query here. And as you can see, I'm mentioning annotation provider is Open Targets. The reason being, we have other providers that provide a target disease association as well, but I'm restricting it to Open Targets currently. If you search this term, of course, it will show you a number of articles. If you select any of those, on the right-hand side, we have list of annotations available for this article. So here you can see it says gene disease Open Targets, and it lists all the target and diseases that has been for which we have found an association. You can select them to go and highlight the sentences where they co-occur, basically. You can also find these associations on Open Target platform. Open Targets platform, on Open Targets platform, you can search for a gene and protein, for example. Here, someone has, I have searched for KIT. It will then provide you a suggestion here at the bottom. What it does is that from the text mined named entities and their associations, it checks which biological entities frequently co-occur and allow you to easily search for those information. For example, from this suggestion, you can tell that KIT actually co-occur very often with imatinib drug, or it has high correlation with colorectal gastrointestinal stromal tumor. So that basically a quick and easy access to further information. So this is my concluding slide. So text mining can greatly help biocuration process. You are most likely already using it, even without knowing it. But there are huge amount of resources available that are not often advertised widely enough. So you probably have to look for them. On the other hand, there are a number of limitations in text mining. To get the most out of this method, you do not need to understand all the concepts in text mining or NLP, but by being aware of some of the challenges, like how we have gene names that are actually common English word. For example, can is a common English word, but we also have a gene name CAN. So, and of course, through text mining pipelines, we try to minimize those false positive identifications, and that can lead to loss of articles that you find. So it's beneficial to know about these challenges, and it will allow you to post-process the data and customize it to fulfill your need, basically. So as I said, if you have any question, and it was by no means an introduction to PMC platform on how you can use text mining on our platform, but do feel free to contact us if you need any help.
Good afternoon, everyone. So my name is Michelle McGrain, and I am the UniProt Curation Coordinator at EMBL-EBI. So I'm going to give an overview of how some of the ways that we're using text mining in UniProt, both to increase the efficiency of the curation workflow and also to improve the information that we're providing to users. So just in case anyone is not familiar with UniProt, so UniProt is a resource of protein sequence and functional information, and it provides comprehensive and high quality protein data. All of the data is made freely available from the UniProt website, which is uniprot.org, and the data is released every 8 weeks. UniProt is an international collaboration. So there are groups at EMBL-EBI in the UK, at the Swiss Institute of Bioinformatics in Geneva, and at the Protein Information Resource, which is at the Georgetown University and the University of Delaware in the US. And these three groups together jointly produce UniProt. And UniProt produces a number of databases. So we have the UniProt knowledge base. We also provide information about protein sets for species with completely sequenced genomes. There's UniRef protein clusters, which cluster protein sequences at various levels of sequence identity, and the UniProt Sequence Archive, or UniPark, which is a non-redundant archive of publicly available sequences from various sequence resources. But our main focus of biocuration is the UniProt knowledge base, so that's what the rest of the talk will focus on. And within the UniProt knowledge base, there are two data sets. So we have the unreviewed Tremble section, where there is no manual review of the data. So in the Tremble section, the protein sequences are imported from various sequence repositories, and additional data is added, which has been generated from computational pipelines. And in contrast to this, in the reviewed SwissProt section, all of the data is manually reviewed, and the data is coming primarily from scientific literature, combined with reviewed results from computational sequence analysis. So obviously, literature curation is of huge importance in UniProt to ensure that important protein data don't remain locked inside PDF. And this process of literature curation ensures that published results are converted into a format which can be both easily understood by users, but is also computationally tractable. And published results are combined with data from other papers and other sources. And this allows us to highlight any conflicting or erroneous data, or to correct data where this is possible, and overall to provide a complete overview of the protein world. So during the UniProt literature curation process, experimental findings are critically evaluated and compiled into a comprehensive report. And the data is represented using controlled vocabularies where possible, and all of the information is linked back to the original publication. And UniProt curators capture a huge amount of information from the scientific literature, from protein and gene names through to functions of a protein for enzymes, what reactions an enzyme catalyzes if it requires, cofactors for its activity, where in the cell a protein is located, in which tissues it's expressed, does it require other interaction partners to be functional, and at the level of the protein sequence, we provide information about things like alternatively spliced forms of the protein, any domains or regions of importance within the protein sequence, an important individual residues, so binding sites and sites which are post-translationally modified, and any residues for enzymes which are important for the catalytic activity of the enzyme. So the curators capture all of this information, pull it together from multiple papers, and add value by structuring it at the same time. So obviously, it's very important for us to be able to easily identify relevant literature. So we want to make sure that the UniProt resource provides information which is as up-to-date and complete as possible, and that the data is of high quality. And also being able to easily identify relevant literature is important for curation efficiency, so that curators can spend less time searching for relevant literature and more time extracting the important data from the literature. So I'm just going to go through some tasks where we have incorporated text mining methods to aid us with these tasks. And the first one I'm going to talk about is our weekly literature triage. And so in UniProt, we monitor the contents of a subset of journals on a weekly basis, and we have incorporated text mining as part of this task. So to do this, we're using Computationally. So Computationally is a tool which has been developed at the NCBI in the group of Zhang Lu. It's a web-based tool, which means that it doesn't need to be installed, and it works across multiple platforms. And it aims to assist manual biocuration by improving curation efficiency. So it integrates various text mining tools, so competition winning tools to recognize various biological entities within the papers. So it incorporates text mining systems for genes and proteins, genetic variants, diseases, chemicals, species, and cell lines. And it provides a PubMed-like interface. So most biocurators are already very familiar with using the kind of interface that Computationally provides. So there's minimal training required before you can get active in using it. And I've given the URL of the tool at the bottom of the slide here. If anybody is interested in digging deeper into the Computationally tool and reading more about how it works, I've given details here of 2 publications which describe the tool, if you'd like to read more about it. So we're using this to monitor a set of 15 journals on a weekly basis to identify new papers. So this includes papers like Cell, Nature, Science, as well as other journals. And the journals are selected based both on impact factor and also species coverage. As I say, we're using Computationally for this task so that we don't have to manually trawl through these journals ourselves. So we are using a customized version of the tool, which has been developed specifically for the needs of UniProt. And the tool sends a weekly email with a summary of results. So it provides a list of these 15 journals that we're interested in and the number of papers in each journal, and also provides a link to the customized interface that we're using. So this is a screenshot of the interface itself. So there's a list of the papers with the entities contained within those papers highlighted. You can also search results by relevance and by date. And there's also a taxonomy filter, which is very useful if you are interested in papers related to a specific organism. And the tool has also been integrated into the UniProt curation editor. So this is a screenshot showing the editor, and we have a tools menu which groups together all of the different tools that are used during the UniProt curation process. So tools that allow curators to do things like run protein sequence alignments or run blast searches. And one of the things that we can do is to launch a Computationally search, and that will launch the tool itself pre-filled with the gene name and the protein name, so that curators can easily access papers related to the particular protein that they're working on. So another area where we have been using text mining is in identifying domain-specific literature. So to identify papers related to a particular area of interest. And to do this, we are using LitSuggest. So LitSuggest is also developed at the NCBI, also in the group of Zhonglu. And it's a web-based system for biomedical literature recommendation and curation. So it incorporates advanced machine learning and information retrieval techniques to find and rank papers that are relevant to a particular area of interest. And again, I've given the link to the tool at the bottom of the slide. And for anyone who's interested in reading more about the tool, there is a recent paper that came out last year. So the details are provided here. And there's also a tutorial provided on the website so that you can go and try it out for yourselves and see how the system works. So LitSuggest allows users to curate, organize, and download results in a single interface. And the results can be fine-tuned by updating the original training corpus that you provide. And you can share your results, so that enables collaborative analysis and curation of the scientific literature. And it provides an automated, personalized weekly digest of newly published articles. Some caveats with LitSuggest are that it only works on PubMed articles. So for example, if you were interested in making use of preprints, then this system doesn't support that. And it also doesn't yet support full text. So it's currently working on abstracts only. So to use LitSuggest, you first create a LitSuggest project, and then you need to build a corpus of positive papers. So these are papers that you know contain data that's relevant to your area of interest. And once you've assembled that corpus of positive papers, you can then use the system to train a machine learning model. And once the model has been trained, then you can produce a classifier which you can run on a corpus of interest. So you could run it over the whole of PubMed, or just weekly updates of PubMed to get new papers as they come out. And so far we have used this to identify papers relevant for curation in two specific areas, which are in curating enzymes and enzyme reactions, and also to identify literature reporting genetic variants. And so we use this to provide a weekly digest of new papers in both of these areas. And it's also been quite successful in allowing us to link existing old data in UniProt to new publications. So this is data where which has been in UniProt for a while, since before we started linking all data to its publication source. So it's allowed us to also be able to go back and provide paper sources for existing data in the resource. And the final area I'm going to touch on today, where we've been making use of text mining, is in expanding literature coverage. So this slide shows a screenshot of a UniProtKB record, and this is looking at the publications view. And when you're looking at the publications related to a UniProtKB record, there's a link to what are called reviewed papers. So these are the papers that were used as the sources of information in the UniProt record. So these are the papers that have been identified and read by the UniProt curators, and the curators have used these papers to extract relevant information. There's also a second link to computationally mapped papers, and these are papers that are imported from other resources. So these are papers that haven't been curated by UniProt. They have been provided by other biological resources, but to provide a fuller picture of the available literature for a particular protein, we have pulled these in from other resources. So this shows the full list of the sources that we are using for the computationally mapped bibliography. So that a lot of these are other curated resources, but we do also make use of some text mining tools to populate this bibliography, and they're shown in bold here. So pGNN is used to identify plant gene names and protein names within the literature, and we're also making use of Computator as well in this process. So this shows what the computationally mapped section of the publications looks like. So all of the automatically imported papers are categorized, and the source of each paper is provided. So in this example here, this paper hasn't been curated by UniProt, it has been provided by Intact. But there's another example here where you can see that the source of the computationally mapped paper is Computator. And this is using the TMVAR text mining tool, which identifies variants and diseases in the literature. So using this method, this paper has been added as it's reporting a variant in this protein, which is associated with a particular disease. And so we've been able to use text mining tools to increase the number of papers that we provide in the computationally mapped section. So just finally to summarize, so our use of text mining in UniProt, it allows curators to find relevant literature more easily, so that makes the curation workflow more efficient and makes it easier for us then to provide a summary of current knowledge from the scientific literature to the research community. And so it allows us additional ways to integrate literature and annotations. So I just want to acknowledge our funders who make it possible for us to do the work that we do, and also to acknowledge the UniProt team. So as I mentioned at the beginning, UniProt is a collaboration between EMBL-EBI, PIR and the SIB, and it's actually our SIB colleagues who have done the work with LitSuggest and training the models and in getting it working with the enzyme and variant areas. And our PIR colleagues have been responsible for the additional bibliography.